membrane. And there are other phospholipids that are on the luminal side. And remember, it's this luminal side that is going to end up, right, on the inside of a transport vesicle, like this, and then facing the outside of the cell when that vesicle merges with the plasma membrane. Right? So there's an asymmetry to these phospholipids, and that gets established in the protein. And that's what we talked about last time. And so I just wanted to check to make sure you remembered some of the names of these proteins that are moving these phospholipids around. So fill in the blanks here with our proper names. One, two, three, or four, guys. All right, you guys are doing well. Most of you picked out number 2. Lipase is one of the ones that is establishing the asymmetry. Scramblase, remember, scrambles everything. All right, so it like randomizes. All right, good. Okay, so we talked a lot about the lipids. Let's talk about proteins. All right, so 98% of our membrane is lipids, but there are proteins there that are doing stuff. And they do different things. And these are sort of some of the major categories of proteins that you'll find in the membrane. So these are like general information of things that you'll find in the membrane. All right, you got things that are involved in transport. So moving things like sodium pumps. That's an example. All right, you got proteins that basically serve as anchors that are holding things by the membrane. All right, you got receptors. These are going to be involved in cell signaling, and we'll have a whole chapter about those guys. So they're involved in cell communication. And then we got, of course, enzymes that are associated with the membrane that catalyze reactions. So these are your big categories. All right, and here are some specific examples. But those are your main categories of proteins. Now, the proteins can be associated with the membrane in different ways. They can be what we call transmembrane, and that means they completely span the lipid bilayer and have part inside the cell and part outside the cell. And they can span the lipid bilayer more than once even. Okay? And these are what we call transmembrane. So they span both sides, both faces of the membrane. All right? They could only be associated with one face of the membrane. They're embedded, in this case, just in the cytosolic face of the membrane. Some proteins are actually linked to lipids that are in the membrane. They're attached to a lipid. All of these kinds of proteins are what we call integral. That means they're firmly attached to the membrane, right? They're either embedded or attached to a lipid. Now, peripheral membrane proteins, on the other hand, these are proteins where if you looked at their location in the cell, they would look like they were close to the membrane. They're not actually embedded in the membrane or covalently bonded to something in the membrane, really. They're associated with other proteins that are embedded in the membrane. And this could be through non-covalent interactions, okay, or outside the cell. Right, it could be with disulfide bonds. It could be broken eventually. Those are peripheral proteins, not actually embedded in the membrane. And the reason why we make this distinction is because if you're studying one of these proteins and you want to isolate it, it's important to know how it's associated with the membrane, because that's going to affect your isolation procedure. If it's an integral protein, you can only get it out of the membrane by using a detergent. We'll talk about those in a minute. All right, so we'll talk about detergents a little bit later. If it's a peripheral protein, which is being held near the membrane by protein-protein interactions, all you have to do is disrupt those protein-protein interactions, and you'll be able to interfere with this interaction, and you'll be able to get your protein that you're interested in free. But the only way you can get these free of the membrane is by breaking up the membrane. Okay, so let's talk about these transmembrane integral proteins. One way for a protein to span the membrane, both faces, is through secondary structures like an alpha helix. We talked about this structure before. This is a really common way for a protein to span the membrane. And if this is spanning the membrane, this alpha helix has to be a certain size, right? It has to be a certain length. And this is usually going to be 20 to 30 amino acids long. And, of course, inside the membrane, right, where these tails are, is hydrophobic. These amino acids, they're side chains in this alpha helix, they are also going to be hydrophobic. They have to like to be in that environment of the membrane. So hydrophilic amino acid side chains are not going to want to be here in the membrane. So the side chains themselves in that alpha helix of those amino acids that are in the transmembrane part of the protein, those are going to be nonpolar, right? Because the nonpolar side chains are the ones that are hydrophobic. Remember that the peptide backbone to form the alpha helix is actually basically hydrogen bonding with itself. All right? The side chains are poking outward, right? Interacting, having hydrophobic interactions with the phospholipid bilayer, and kind of protecting the peptide backbone, which is interacting with itself. So our peptide backbone is kind of like folding up in on itself in that alpha helix, and our side chains are poking outward, protecting it from that hydrophobic environment. Now, if you have the amino acid sequence of a polypeptide, you have information, because we know the properties of the side chains of the amino acids, right? We can use computer programs to tell us if there are things like hydrophobic or hydrophilic regions of this polypeptide. And that is what this kind of graph here is telling us. This kind of graph is called a hydropathy, or sometimes it's called a hydrophobicity plot. And it can be used to predict transmembrane regions of proteins. Okay, so in these graphs, what you're looking at here on the x-axis, this is for two different polypeptides. So this is polypeptide 1 over here, polypeptide 2 over here. You're looking at the amino acid number in the sequence of the primary sequence. And then over here on the y-axis, you're looking at what's called the hydropathy index. Right? How hydrophobic is that particular amino acid? All right. If it's a positive value for this kind of analysis, that's going to indicate more hydrophobic segments of our polypeptide. Okay. Now notice for our two polypeptides, the first one is glycophorin. What we have at the top here is like a 2D diagram of the polypeptide. It's showing us here is the N-terminal region, then we have some amino acid sequence, and then we have the C-terminal region. And this diagram is showing us right here, right? This is where this peak is. Right? And these other regions are these troughs. Okay? So this region right here corresponds to this, and this is the hydrophobic region. This could be a transmembrane region of this protein because it's very hydrophobic. All right, here, same analysis, just a more complicated polypeptide. The longer polypeptide, and it's got more peaks in it. It's actually got 7 peaks. Could have 7 transmembrane regions. And so here's the 2-dimensional diagram of that polypeptide. Question about the hydropathy plot. Yep.
So you said the positive sides are for more hydrophobic properties.
Yeah, so when it's above 0, that's indicating a hydrophobic segment of the polypeptide sequence. Other questions? Okay. So we were talking about alpha helices. It's also important to realize that beta pleated sheets can also form transmembrane domains. So this is showing you guys how a beta barrel can form using beta pleated sheets. All right, and so these can form really large transmembrane channels. So basically you have the beta pleated sheet that is basically forming like a big tube. All right, so these are going to have, of course, hydrophobic side chains. All of these amino acids that are here are going to have hydrophobic side chains. So you can have other secondary structures that are able to fold up, and if they're hydrophobic, they can form one of these structures that can span the membrane. But alpha helices are more common. Now, you don't have to have just one, you can have multiples that form complicated structures. So here, we've got 5 alpha helices. Instead of looking at ribbons, we've kind of drawn them as tubes. And these are forming, basically, a channel. And so the way that these alpha helices have their structure is that all of the amino acid residues here on the outside in green, those are hydrophobic amino acids. But the ones that are facing the center, these are hydrophilic. So they're able to interact with each other and also the cytosol. So they're basically making a space in the membrane that cytosol can pass through and make a channel. So depending on how these secondary structures fold up, right, you can get really complicated than tertiary structures in the membrane. do, if this is the 2D diagram of a polypeptide, and this over here is the N-terminus, and this over here is the C-terminus, and the dark blue represents transmembrane regions. How many peaks is this polypeptide going to have on hydroxy block?
Tree.
So you guys can show me. Pick a number, right, of the answer. Good. Right, 3 peaks above 0 because you have 3 transmembrane regions. So it's going to have 3 peaks. All right, that part's pretty straightforward. All right, try this. All right, here's a ribbon diagram of a transmembrane protein. All right, this region in here, all right, this is the phospholipid bilayer. I want you guys to draw 2 things. Draw the linear schematic, right? Like we just predicted how many peaks there were from that linear schematic. Draw a linear schematic.